This video summarizes the poster presented at the America's Committee for Treatment and Research in Multiple Sclerosis Forum 2023. The title of this poster is Chronically Expressed Blood-Borne Endogenous Ancestral Nucleotide Sequences, ENS, RNA of patients with multiple sclerosis, Alzheimer's, or Parkinson's disease are biomarkers for differential diagnosis and monitoring. You can get a copy of this poster by directly contacting ACTRIMS on their website, actrims.org. The reason why we did this experiment was to test the hypothesis that mankind is in fact a mosaic of endogenous ancestral nucleotide sequences from endogenous retroviruses to hominids. We know this from last year's Nobel Prize for the fact that we have 3% of our genes are from Neanderthal. And so the purpose of this study then was two objectives. The first objective was to quantitate a blood RNA biomarker of patients with neurologic diseases as compared to apparently healthy controls. The second objective was to follow 3 relapsing and remitting multiple sclerosis patients over time to determine if there are any correlations between blood RNA biomarker expression and clinical observation. The methods that we used was to take retrospective samples from a vendor that had frozen PAX gene tubes of multiple sclerosis patients, Parkinson's disease, patients with mild cognitive impairment, and apparently healthy controls, and to extract the RNA and to look for these ENS and measure them in the various different groups. We then used artificial intelligence and machine learning to be able to then give us various different measurements, including PCA plots. The way we measure the expression of ENS in the blood of patients with various diseases was to extract the RNA and then specially treat it with an FBB reagent that enriches for endogenous ancestral sequences. And once we have those sequences, we ask, what is their expression? This graph illustrates the power of artificial intelligence analysis of the FBB Biomed enrichment process. We can differentially diagnose patients with multiple sclerosis, Alzheimer's disease, Parkinson's disease from each other and from apparently healthy controls. In this small study, you can see how each group bundles together. Let's take a look at figure 1, where here you see a treatment responsive patient over 7 visits, each one spaced apart by 6 months. And you can see that the patient shows a decreasing amount of one of its biomarker panels to a point of very extreme lowering of the variance as a result of steroid transfusion right before the measurement was taken. Whereas the second region of interest had not as much of a response. This is important when one is gauging what kind of drug therapy to use for each patient, and especially when combination therapy may be needed. The second figure shows what happens when a patient came off a research study on copazone and then slowly over time and finally the third visit started to show positive variance in one of the regions of interest to a point that almost all but one visit had a flare showing the patient became non-responsive to drugs over time. And figure 3, you can see here that the variance is always over the 0 line, and this patient basically had flares except for 2 points throughout the baseline of the study. So in conclusion, diagnosis of neurologic diseases remains difficult and time-consuming. The FBB Biomed enrichment protocol and the proprietary ENS reference database were used to analyze RNA-seq data to discover specific biomarkers that assist in the definitive diagnosis of neurologic diseases. The ENS database recognizes the genomic mosaic basis of humans. Matching FBB Biomed's proprietary regions of interest with bioinformatic analytical tools such as principal component analysis allows us differential diagnosis. The chronic expression of some of these unique genes appears to prognosticate with symptom exacerbations and could be used then as a companion diagnostic. And lastly, we think more studies should focus on the reactivation of endogenous viruses. The reactivation of latent endogenous viral-like elements should be considered a likely source for viral-associated antigenicity. The activation and chronic expression of genes in neurologic disease patients forms the basis of more in-depth investigations into the pathologic mechanisms of activation of ancestral sequences and their role in neurologic diseases.